I'm David Eveleth, CEO of Trefoil Therapeutics. We're focused on corneal disease and developing therapeutics for corneal disease because if you have a corneal disease today, you're likely to experience visual impairment and your therapeutic options other than surgery are limited. Trefoil is using its engineered FGF1 technology to regenerate corneal tissue and restore sight without surgery. We're developing products for both the back of the eye, back of the cornea, and the front of the cornea using intracameral and eye drop formulations. We're developing two first-in-class products for front and back of the cornea indications, attacking diseases for which there's no current pharmacologic treatment that have an aggregate revenue potential on the order of $2.5 billion. And our next value inflection point is the conclusion of a clinical proof of concept study in Fuchs dystrophy and the filing of an IND in a front of the cornea indication, both in 2021. Our first target is corneal endothelial dystrophy, which is the leading cause for corneal transplantation today. Corneal endothelial dystrophy is a degeneration of the cells that line the inner surface of the cornea, leading to the accumulation of guttae, corneal edema, corneal opacity, and all these things impair vision. The current therapeutic approach is the surgical transplantation of donor tissue into the inside of the cornea, a process that's effective, but it's surgically challenging, it's expensive, and it carries with it the requirement for lifelong steroid use with the attendant adverse events and the potential for the rejection of the transplanted tissue. Trefoil's drug is aimed at regenerating the patient's own endothelial tissue and thus restoring vision without surgery, avoiding the need for lifelong steroid therapy, and eliminating the possibility of the rejection of transplanted tissue. But corneal endothelial dystrophy goes beyond just Fuchs disease. The number of your endothelial cells on the inside of your cornea decreases with time. And so there are about 12 million patients out there with corneal endothelial dystrophy. Only about 600,000 of these patients are diagnosed, and only about 200,000 of those will go on to have corneal transplantation within the next 2 years or so. But even patients that are mildly affected are impacted by the disease because they can be rejected from cataract surgery because of the risk of complications from doing cataract surgery on a patient with corneal dystrophy. And the number of these patients continues to increase as the population ages. We believe that the availability of a more cost-effective therapy that can be applied earlier in the disease will increase both the treatment and the diagnosis rates. So we looked at the possibility that our drug could cause corneal endothelial cells to proliferate in corneas taken from Fuchs patients. And on the left, you can see a control cornea, and in the middle, a cornea that has been treated with drug, with the green dye marking the cells that are proliferative. And you can see that the drug increases the proportion of cells that are proliferating, and looked at quantitatively on the right. Our drug induces a 4-fold increase in the proportion of cells that are proliferating in the endothelial layer in these corneas taken from Fuchs patients. In short, we have multiple validating efficacy studies. We can make corneal endothelial cells proliferate in vitro. We can accelerate the clearing of the cornea in a rabbit cryo-injury model, and we can make corneal endothelial cells in corneas from Fuchs patients proliferate. On the front of the cornea, these diseases are characterized by defects in the epithelium that lead to corneal opacity, haze, and pain. And our focus here is herpetic keratopathy and ulcerative keratitis. On the left, you can see that our drug induces a significant decrease in corneal edema in a rabbit model of mustard gas poisoning. And on the right, we induce a significant decrease in the amount of keratitis in a mouse herpetic keratitis model. Taken together, these two indications represent about a $2.5 billion revenue opportunity, with the Fuchs dystrophy and associated surgical indications representing about a $1.7 billion opportunity, and herpetic keratopathy and ulcerative keratitis representing about an $800 million opportunity. Trefoil is currently seeking a Series A financing to take it to the next value inflection point, which is the completion of our clinical proof of concept study in Fuchs and the filing of an IND in front of the cornea indication. This will position us to begin two pivotal programs in two different indications. Our core management team has decades of experience in the development of ophthalmic drugs. In summary, we're developing two first-in-class products for the front and back of the cornea with an aggregate revenue potential in excess of $2.5 billion using conservative market share assumptions and with demographics driving growth. We have an efficient development plan that will enable us to reach two proofs of concept in two different indications in 3 years. And we have an experienced management team, the management, the board, and our advisors that have an established and successful track record in the development of ophthalmic drugs and in biotech exits. Thank you.